Hello, today we are going to continue talking about different types of sequencing projects, and I'm going to specifically focus on RNA sequencing or RNA-seq in this video. As a reminder, here's a diagram of a eukaryotic cell. You have a specific cell type that I've drawn here in yellow. The nucleus is drawn in red, and the blue DNA is housed in the nucleus. RNA is transcribed from DNA in the nucleus and transported into the cytoplasm of the cell, where it is translated into protein. A couple things that you might immediately notice about RNA is that not all of the DNA, not the entire genome, is transcribed to RNA, and whatever is transcribed to RNA theoretically has a role in whatever proteins are being produced in that cell. This is why RNA sequencing is valuable to genomicists and to all biologists, because by sequencing RNA, we can get a sense of the biology of this entire cell. Depending on what type of cell this is, what type of condition that it's in, it might be expressing different RNAs or different amounts of certain genes in order to control the proteins that are being produced and the biological function of that cell. How do you do an RNA sequencing project? Or from now on, I'll just say RNA-seq. The first step is to start with either a population of cells, that is maybe an entire tissue worth of cells, or today we're getting to where we can we can isolate RNA from even a single cell to do an analysis. So whatever you're starting with, a population of cells or a single cell, you will have everything that's in that cell, right? You'll have DNA and cell membrane and proteins and RNA. And by some mechanism, you want to isolate RNA so that at the end of that, you have a tube that just contains your RNA. The next step is to prepare your library. A library is all of the RNAs that you have modified such that they can be read by a sequencing machine. That's why we call the result of our sequencing effort the reads. And we say, how many reads did you get back from the sequencer? And then how many of these molecules did the sequencing machine find? So yeah, I'm being very vague in all of these steps because it is not my intention right now to show you one specific protocol for RNA-seq. There are many protocols for RNA-seq, but it is important that you know that usually this involves adding some sort of adapters, some sorts of sequence to either end of the actual RNA that can allow this read to be read by the sequencer machine. And this is true of all sequencing platforms. I've been glossing over it a little bit, but it doesn't matter if you're sequencing DNA in a whole genome sequence or you're doing a RNAseq project. You will have to prepare these nucleotides in some way such that they can be accessed by the sequencing machine. The next thing you want to do with reads that you get back from a sequencing machine is trim them. That is, maybe get rid of those adapters or any other sequences that you stuck on them that should not map to your reference genome or your reference transcriptome or whatever it is that you're using. Get rid of those and also do a quality filter. So we talked about how sequencing machines don't produce 100% confident sequence data. Sometimes they're just not sure about a particular base, or sometimes even an entire read is just kind of crappy. So this is the step where you would get rid of these. So now you have all of your RNAs that have been trimmed of adapter sequences or any other intervening sequences and are only the highest quality. And the next step will be to map to your reference. Here I'm going to demonstrate this next stage of mapping to a reference genome, which I have depicted here in blue. And this is, let's just say, part of a genome that contains 3 genes with regions in between those 3 genes, gene A, gene B, and gene C. So our task now is to take all of our RNA-seq reads, which I have in green, and map them to this reference. So what do I mean by map? I mean find where they align. So you take this sequence, or your program takes this sequence, and it says, does it fit here? Does it fit here? Does it fit here? Because remember, I have just drawn these as squiggly lines, but on that squiggly line, you should imagine is A's and T's and G's and C's of a sequence. So where does that sequence match? So let's say that this read matches here to gene C, gene B, gene A, and let's say one more at gene B, if I can grab this one. He's being stubborn. Okay. And because this is RNA-seq, the number of times that we see a read of a particular gene is a reflection of how many copies of RNA were in that cell for that gene. We should also remember that because we're looking at RNA, when we're mapping these RNA-seq reads to the genome, we might encounter regions where there are introns in the reference genome, but not in our processed RNA transcript. So, depending on the type of library you prepare that may or may not um, have long enough reads that might contain an intron, when you're mapping, the actual result of your mapping might be something like this, where part of your read maps to the reference genome, part of it doesn't. That's putatively an intron, and then the remainder of the read maps to somewhere else along the genome. With this type of RNA sequencing, you may even be able to detect alternative splicing. I've drawn another transcript here, which we see mapped to gene C, but in this transcript, the read we got only had what is depicted in this diagram as, say, maybe exon 1 and exon 3, whereas the other version of the transcript that we saw in our reads, therefore it must have been present in the cell, has all 3 exons retained in the final transcript. So if we see instances like this, where maybe only some of the exons are retained versus all of the exons retained, this type of technology can provide evidence of alternative splicing. I'm going to return to our simpler diagram of the reference genome with reads mapped to it. You can pretend that there are still introns in these reads that we are just not depicting. And I'm going to show you another real power of RNA-seq, which is differential gene expression analysis. So because the number of reads that you get for each gene is related to the number of RNA copies in that cell, it can tell you something about the physiology of that cell. And you can compare it to a different state. So one example that I'm showing here is imagine that you're comparing liver cells, the types of genes that are expressed in tissue that you would get from a liver, from lung cells, for example. And based on which reads we saw and how they map to the genes, you can make statements about differential expression. So this stage of the analysis is what we would call counting. So you're counting how many times do I see each of these transcripts for each of these genes in each of my conditions or cell types or whatever it is that I'm comparing. So what I would take away from this analysis right now is that gene A is upregulated in lung cells relative to liver cells. Gene B is not differentially expressed between liver cells and lung cells, and gene C is upregulated in liver cells with respect to lung cells. So this type of biological question is a little bit different than what we've been addressing so far, because while variants may exist in the sequence here, this mapping doesn't have to be perfect to the reference genome, by the way. We've talked about this a little bit. There may still be a one base pair substitution in this read. For the most part, when you're looking at RNA-seq data, you're not usually concerned with detecting sequence variants. You're concerned with detecting transcripts, that is, maybe seeing which exons are present, what alternative splice options are available in the cell, and you're also interested in comparing the expression of different genes between different conditions. So I'm going to stop for now, but hopefully that has enhanced our ability to design genomic techniques by introducing this new technology.